Our next speaker is Kirk Beebe. Dr. Beebe is the Director of Application Science at Metabolon. His experience includes driving forward the science of the metabolism of proliferative cells and the application of metabolomics to as a comparison for advancing cell culture processes. He holds a PhD in biochemistry from Ohio State University. I went to the University of Michigan, and was a postdoctoral scientist at the Scripps Research Institute. The title of his presentation is Employing Metabolomics in Cell Culture and Bioprocessing to Gain Greater Predictability, Control, and Quality.
Thank you. Let me go back here. Yeah, well, that's all right. Our CSO's from the University of Michigan too, so you know, there are a lot of battles, particularly this time of year. So today I'm going to take a step back and talk a little bit about how you can use this metabolomics technology in any array of areas, whether it's trying to produce a stem cell-like product where the cells are the product, or whether it's an antibody. Essentially, it's a technology that can allow you to open up the hood of any biological system and begin to understand it a little bit better. The whole purpose of understanding, ultimately, is to have better predictability of the system, control, and ultimately quality as well. And I see this afternoon's session will be a lot of discussion about quality, and I think the one case study I have selected today has big implications with respect to quality by design. So if we just start here with just a basic cartoon here, I think a lot of people who work with different cell lines, or certainly stem cells, will admit to some degree these are incredibly complex. There's so many variables. We're still dealing with a bit of a black box system, particularly with respect to how do we actually go between the this very complex cellular system into what are all the factors that conspire together ultimately to produce this kind of high quality product in a vial. And really, if you take a step back from it, what are the factors that limit that? It's really just having enough knowledge, and then also having the biomarkers or the signatures that can inform you about what's going on underneath the system. So it's really a combination of building knowledge infrastructure and biomarker infrastructure to begin to deconstruct more of this. And essentially, if you zoom out from that concept a little bit, it's helpful just to think about, well, what are the main kind of driver components of what goes on underneath this? So what are the main components that drive this cell, let's say, to ultimately produce high quality product in a vial? And really at a very, very high level, this is fairly straightforward, but it's the genetics of the system. We all understand that very well. The environment of the system, we understand that as well. And I'm not telling you anything you don't already know. It's also what we feed it, or the media. But we really don't have a single kind of diagnostic tool to allow you to incorporate all the information and the drivers here into what actually is driving a cell ultimately to produce this product. So we don't have a way to really incorporate, is it an environmental, a nutritional, or genetic component that's actually driving this? And that's where I think the use of metabolomics actually comes in. Because if you think about measuring metabolites, so basically these small molecule end products of the central dogma starting with genetics, metabolites actually are essentially a surrogate of the phenotype. So if you have environmental changes, and I'll show you that with the one case study, you'll very frequently see metabolic responses to that. Same thing with nutrition when you're feeding it certain things. So we saw that to some degree in the last talk, that there are certain elements, in this case antioxidants, that can infect the ultimate phenotype, which is the production, in this case, of a slightly different coloration of the antibody, or the ultimate, or the end product. And then so in essence, with metabolites being a surrogate of this phenotype, you can account for environment, nutrition, and then also the genetics very nicely looking at metabolites. Now, it's not trivial to do that, but ultimately, if you're able to develop a system where you can capture this data and then effectively analyze it, you can gain a lot of information from it. So this just gives you a bird's eye view of what we think of when we think of metabolomics. So we're not talking about measuring just a few analytes, 10 or 20 analytes. We're talking about trying to take a cell pellet or a media sample or tissue and measure several hundred analytes in one shot. So basically, this is a large kind of wall chart of biochemistry displaying pathways in lipid metabolism, amino acid metabolism, energy, and so forth. The idea is that we've built a system that can survey this very broadly. So in essence, we can try and capture what's the metabolic profile underneath the system that's driving a particular phenotype. And one quick thing just to note here as well, when people think of metabolomics or metabolic profiling, very often there's an immense amount of focus on this step. And it's a critical step, so that's where you might have the underlying analytical chemistry. But it's really it falls short in really being able to deliver something useful in the end. This is a critical step here, being able to take the data and process it and QC it rapidly. And then the other thing that once you've created a high quality data set, the other thing that's really important is just having designed the study appropriately, such that the end result matches. You know, the study is designed such that for the study goal in mind. And then the last little thing is that once you produce that high quality data set, the other thing that often isn't really is viewed as being somewhat trivial is that you really have to be able to interpret the data. You have to build a system to extract information from the data as well. So that's kind of what, just to give you a bird's eye view of how we view metabolomics, that's really kind of how we view it. It's a multi-step process. It's not just about capturing data. So in essence, what we view is that metabolomics can be this surrogate in essence. It can actually be the diagnostic data stream that can help you understand a little bit more how you get from this complex cellular system into a high quality product. Because again, it can take into account the genes, the environment, and then what we're feeding it. So a lot of what I've already told you is probably, you might be saying, well, yeah, we already, this is something we already do, and that's a good thing when we're talking to people who are interested in cell culture. But this is just an extension of what's already done. Obviously, people in a lab typically are profiling routine metabolites like ammonia, glucose, a few select amino acids, lactate. And what this typically does is it captures a lot of information. It's very, very useful, but I don't think I often have, or I haven't ever really encountered an argument that this doesn't really completely survey phenotypic space. Otherwise, there'd never be any surprises. These things would always work. There'd never be any issues. So what we're suggesting really is that you could just use this type of technology, this metabolomics technology, to begin to survey this space more broadly. And not necessarily survey it every time hundreds of metabolites, but survey it in meaningful experiments to essentially design those next assays, basically develop those next signatures that you can then pull into a more targeted setting and a more routine use that can inform about certain cell culture phenotypes. So that's the basic premise, using this technology to find, let me go back here, using this technology to find those new signatures that can be used in a more routine basis. So I'm going to dive into a specific case study in a minute, but just to give you a sense, a little bit more of a bird's eye view of how many people have used this technology. This is just a quick summary of some typical objectives and outcomes of different studies. And it does generally fall into the bin of people trying to improve quality, trying to improve productivity, and then just basic troubleshooting or biomarker infrastructure. So I'm not going to talk about this one today, but many people are interested in just trying to improve product quality, whether it's glycoforms or whether it's some sort of disulfide heterogeneity. And often will use this technology trying to understand what the basis of that is. And then once they use metabolomics to understand it a little bit better, they can then devise strategies to improve it. So that's quite common. I'm not going to go into a lot of details with either of these cases today, but I will allude to a very similar case in the main case study I'm going to talk about. The other thing is just simply trying to gain better control or improve productivity. Very often this might be with standard workhorse industrial cell lines like CHO cells, but it can often also be in other non-mammalian hosts or microbial systems. And the idea there is, again, use the technology to look under the hood of the system to figure out how you can improve the process or the media. And lastly, there are many people that are using this to build biomarker infrastructure. So in other words, the last talk was a good example of developing new assays or endpoints that can be more informative than old assays. Well, it's the same thing here, whether it's something as advanced as finding a biomarker that can help you predict if something will go from a shake flask to a bioreactor more effectively, or a biomarker that can be more informative in a dynamic feeding algorithm. The idea is building biomarker infrastructure. So the one example I'm going to show you, and I might have time to allude to a second one, but is a classic case of looking at a CHO cell process with a monoclonal antibody. And if we start up at the top here, this all started around a question of pH. So as they were doing process characterization, they noticed that this particular process cell line combination was particularly sensitive to pH. So yes, we all know if you change the pH, you often change the way the cell culture process will behave. But in this case, a very, very small change resulted in a dramatic phenotypic difference, which I'll show you on the next slide. So they, in the interest of building knowledge infrastructure, they said, well, let's try and understand the basis of this. Let's try and understand what are the underlying components that are driving these changes. So it was just simple. They set up a process with a control and the different pH set point, and they just profiled the cells and media over the various process days. So, and then we essentially took extract of the cell pellets, extract of the media, put it across the metabolomics platform, and measured several hundred analytes. So this is just to give you an idea of what the main metabolic difference was between these two conditions. And this is a classic challenge you see in any cell culture process, that as they're proliferating rapidly, they're producing a lot of lactate. You can see both of these conditions, the control and the higher pH set point, were producing lactate as you'd expect early on. But whereas the control exhibits this nice, well-behaved pattern that as some degree of confluency is reached, it begins to, there's a cessation of production and consumption of lactate. You see that the high pH set point just continues on at a very high trajectory. So the basic question was, and this was the kind of the pinnacle of what they were trying to understand in the study, is what was the underlying basis for this. And the other thing I should comment on is there was really only one other major phenotypic difference here, and it was that there was a product quality difference between these two. It did produce a product quality difference. And I might be able to get into that a little bit as we go on. So again, they profiled the cell pellets and media across this, and this is just to show you that in both the cell pellets and the media, when you just look at, this is just looking at ratio comparisons between the control and the high pH set point, you see a lot of metabolites were measured across the process days, and there were a lot of differences as shown by the red and green. But if you actually look at the data very closely and you ask the question, what were the most significant changes that occurred at the point of divergence, at the point that the two phenotypes clearly diverged? What you find is there's really only about a dozen metabolites that actually show a very clear pattern, particularly in the cells, at the point of divergence. So just to orient you briefly, this is just a heat map across the process days, and here are some of the analytes. And if you see a red shaded cell, it just means it went up in the high pH set point. In green, it went down. And what you see is a number of analytes here that exhibit this pattern, which is essentially the control remains fairly flat and the high pH set point increases and stays somewhat persistent across that time. So most of these analytes, other than lactate being one of them, most of these you can actually assign to being involved in osmoprotection. So like glycerylphosphocholine shown here is a major organic osmolyte. So that's not necessarily surprising to see this, given that there's a higher pH set point. But the reality is you can have high pH set points, and you can even have changes in osmo and not always see this response. This particular cell line process combination was particularly sensitive and was actively exerting an osmoprotective response. So obviously the concept was this is the clearest signature that's associated with the lactate What could this indeed be osmo? Well, if you look at another class of analytes, so this is just looking at all 20 amino acids in the media in this top block here, and then the cell pellets. And I just took a couple of examples out here as plots of various amino acids. But basically what you see here is there's a pretty distinct pattern here. In essence, you see that very early on, the high pH set point, there's an increase in amino acids in the cells. They begin to take up a lot of amino acids. And as a consequence, you actually see that by the green here, that they're actually running out of amino acids by the end. So this has obvious, if nothing else, this has obvious media development implications. But it also partially explains the product quality issue we saw as well. And I'll show you down here. See this little strip of green right here? That's an amino acid that's running out, and that happens to be cysteine or cysteine. So basically, it's running out of a precursor for making glutathione. In fact, what they were able to do is actually supplement that condition with this and show that you could improve the product quality because of that. But this all pointed to the cells responding to osmo protection. Indeed, if you look at the osmo, the profile is very, very similar to what you saw with some of those metabolites responding. And then the question really turns to, okay, great, we seem to see that indeed we have a higher pH set point, there's an increased osmo, the cells are actually mounting an osmo response, but how is that actually relating back to the lactate phenotype? How is it producing this metabolic response? Well, when you look at the data further, you can find that there are a number of metabolites. So this is a block of metabolites in the media, and this is in the cells. And again, red is increasing with time, green is decreasing. And basically what you can see is a number of metabolites to indicate there's a impaired mitochondrial function. Organic acids, TCA cycle intermediates, basically carbon that's not being effectively consumed. And probably the poster child for this might be pyruvate that I show here. That you can see again in the control condition, there's a little bit of a bolus in the beginning, but it's basically gone by the end of the run, which is what you'd expect in a well-behaved process. But yet the pyruvate is much higher in the media in this other condition. So it's really indicating that there's a impaired mitochondrial catabolic capacity, and this was confirmed by a separate independent assay. So you went from just having an observation of high lactate production to understanding it a little bit better and being able to derive a model that goes like this. In essence, initially, as you'd expect with cells that are proliferating rapidly, you see that they're, through glycolysis, producing a lot of lactate, lots of lactate being produced in the control and the high pH set point. But in the high pH set point condition, there's a more aggressive response to pump base in, right? So that creates a hyperosmotic state. Then this hyperosmotic state, actually, we propose is suppressing mitochondrial function and causing even more lactate production. And what you can see is the cells begin to adapt in different ways. They adapt to this by taking up amino acids for osmo protection. They also produce these osmolytes like glycerylphosphocholine. So they actually begin to adapt. And you see that, we propose that you see some partial restoration because you do see the lactate begin to come down near the end. But indeed, these cells are particularly sensitive to osmo differences. So this just helps to build a model and begin to build more knowledge infrastructure of a particular process. So understanding something like pH is expanded a little bit. The other thing that's interesting about this is, again, if you go back to what I mentioned about not all cells that have to grow in higher pH or even higher osmo will respond this aggressively. So how to identify the ones that will, other than the overt, there's a lot of lactate being produced, which could be due to a number of different reasons. Well, one of the things that could come out of this type of study is that you could use something like glycerylphosphocholine as a biomarker that the cells are actually responding, and they're particularly sensitive to osmo differences. The kind of practical implications from this study, besides just knowledge and biomarkers, is that you could clearly see that if you were to look at the media and cells in such detail, let's say you had to operate under different process conditions, you can clearly see there are different media and feed strategies that would have to take place. For example, the cysteine that I mentioned, that again, the depletion in cysteine, when it was added back, you could improve the product quality of this particular condition. And that kind of shows the just general utility of being able to look under the hood of the system with this. And I think that's about, I did have one more example, but I'm going to skip ahead because it's just a summary anyway. And I'm going to wrap it up here just since it is almost lunchtime. So acknowledgments, this work was done with folks at Pfizer, and there was some other work I didn't have a chance to talk about with folks at the Keck Graduate Institute, and then my colleague Doug Lee at Metabolon. And hopefully the idea here is you get a sense that this is technology, whether it's stem cells or whether it's antibody-producing cells, that can allow you to open up the hood of a biological system and understand it a little bit better, gain biomarkers for being able to predict certain phenotypes. And I'll just leave it at that and take questions.
Questions? Yeah. I had one actually.
Yeah.
Can you describe what sort of product quality issues you saw in the high pH samples?
Yeah, that happened to be directly related to the low cysteine levels, which led to low glutathione levels. So it was disulfide heterogeneity. Yep. Yes?
You said talked about low cysteine levels. Was that in the cell pellet or was it in the supernatant?
Okay, so actually, very good question, because if you looked in the media, you did see that it was lower in the media, but the cell pellet is where you really saw the differences, because you saw glutathione levels and cysteine levels in the cells actually deplete. The concentrations of cysteine were different in the media. But the way you could really confirmed it was looking in the cell pellets, where you saw not just lower levels of cysteine, but you also saw glutathione levels almost mirror that identically. So it's nice in these type of studies is to try and look at the cell pellets and media, because I've done a lot of studies where the media gave the key clue or the cell pellet gave the key clue, but oftentimes they dovetail nicely.